Hello, I am Sumedha Jayasena. This presentation is meant to give you some updates on the development of vaccines against the SARS-CoV-2 virus to control its spread around the world and COVID-19, the disease caused by this virus. The dawn of the cold weather in the winter and the upcoming holiday season provides a fertile opportunity for this virus to spread at an alarming rate, which we have already begun to notice across the United States. If we don't get a vaccine on time, the virus will infect hundreds of millions of us and causing millions of deaths. As of November 22, 2020, there are 58.5 million infections and 1.3 million human lives lost. A sobering figure. We must also understand that more than 98% of infected individuals will recover from COVID-19. If we fail to control the virus spread, it will infect human populations until it reaches 67% of the people infected. At that point, the community has acquired herd immunity to protect the remaining 23% of uninfected individuals. Without letting the virus to infect us, we could achieve herd immunity through vaccination. For many reasons and beliefs, some of us are opposed to vaccination. It is a personal choice. However, they must understand that they are also being protected by those around them who receive the vaccination. The requirement of a vaccine is to bring our lives to some degree of normalcy as soon as possible. To develop a vaccine against the SARS-CoV-2 virus, several billions of dollars have already been invested. Let's start with the natural principle that almost all living organisms adhere to. That is the transfer of genetic information stored in DNA. The information stored in DNA is copied or transcribed to RNA. This RNA is called the messenger RNA or mRNA, as it carries the message stored in DNA. The information copied to mRNA is translated into a protein molecule. The protein molecule interprets the code stored in DNA and executes the function. This information will help you understand the rest of the presentation. SARS-CoV-2 is an enveloped positive strand RNA virus. The outer layer of the envelope is derived from the membrane of an infected human cell. Inside the envelope is the viral genome, which is a positive strand of RNA. Positiveness indicates that the viral genome is ready to be translated into viral proteins as soon as it infects or enters a human cell. Out of several viral proteins, one in particular sticks out of the viral envelope. This protein is called the spike protein or S protein. The S protein is the key to infecting human cells upon engaging with the ACE2 receptor found on the surface of human cells. Cells that carry the ACE2 receptors are abundant on the surface of our lungs and the upper respiratory system. Once a virus enters a human cell, it hijacks our protein synthesis machinery to produce viral proteins. Some of the viral proteins help replicate the viral genome to create daughter viruses encapsulated in the human cell membrane. These daughter viruses then escape the infected cell to infect many other cells, thereby establishing COVID-19 disease. Our immune system is one of the most complex biological systems. It consists of an array of cell types, proteins and chemicals. This presentation mostly restricts to a class of proteins called antibodies. Antibodies play a crucial role in our immunity. In a very general sense, when we talk about the immunity, what immediately comes to our mind is the word antibodies. Immunity comes in two forms. The first is passive immunity. Passive immunity is given to us by someone else. That means it is transient and does not last very long in our body. Passive immunity can come naturally from the mother to the developing fetus in her womb. She sends her own antibodies through the placenta to protect the fetus. Once we are born, our immune system is not fully developed, and we still need help to protect ourselves until we develop our own immune system. Again, our mothers protect us by providing antibodies through her breast milk. Colostrum, the transparent portion of breast milk, contains the mother's antibodies. Alternatively, we could artificially receive passive immunity. For example, the blood plasma of a patient who recovered from a viral infection contains antibodies that he or she used to fight the infection. This plasma is called convalescent plasma. Convalescent plasma obtained from recovered COVID-19 patients is used to treat severe COVID-19 disease in the clinic. An antibody is a Y-shaped protein molecule consisting of two binding sites recognizing a unique portion of another molecule. For example, an antibody could recognize a unique portion of the S protein of the SARS-CoV-2 virus. By doing so, this type of antibody can block the viral infection by preventing or blocking the binding of the S protein to the ACE2 receptor on human cells. These antibodies are called blocking or neutralizing antibodies. To treat COVID-19, two companies have produced neutralizing antibodies that block the viral entry. The first being the pharmaceutical company Eli Lilly. They manufactured a neutralizing antibody by antibody engineering. The second came from Regeneron, a biotech company that produced an antibody cocktail containing two neutralizing antibodies derived from a convalescent plasma of a COVID-19 patient. This is the cocktail that was included in the treatment regimen of President Trump when he got COVID-19. In November 2020, the FDA approved the antibody products of both companies for emergency use for COVID-19 patients. These treatments cost between $1,250 to $1,500. The efficacy of these neutralizing or blocking antibody treatments is transient, and it only lasts as long as the antibodies remain in the body, which is less than a month. This treatment helps reduce the viral load and the severity of the COVID-19 disease. The second type of immunity is active immunity. We build the active immunity within our body. One straightforward way of building immunity against a virus is, in fact, getting infected by the virus, a natural infection. For example, having chickenpox infection during our childhood builds lifelong immunity against the chickenpox virus. If you are old or have underlying health issues like diabetes, heart, or kidney diseases, getting infected by the SARS-CoV-2 virus is not a good option. Instead of getting a natural infection, we could build or induce active immunity using a vaccine. Over the past 2 centuries, vaccines have saved several billions of human lives from deadly pathogens. Vaccines help eliminate deadly pathogens from their existence. An example is the smallpox virus. Vaccines train our immune system to recognize and control an infection caused by a pathogen like a virus. Instead of the disease-causing natural virus, in a vaccine what we show is a portion of the virus or an inactivated version of the virus to our immune system to train on to combat the natural virus later when it is presented. Vaccines are produced using different platforms or processes. Historical examples of vaccines produced by different platforms are shown here. Whole cell vaccines come in 2 forms, either live attenuated vaccines or inactivated vaccines. A live attenuated vaccine is produced by growing or culturing the natural disease-causing virus under conditions that are not suitable or permissible for its growth. For example, culturing in animal cells or growing it under low temperature. These live attenuated viruses, when given in a vaccine, could replicate at a much lower rate inside our body, but they cannot cause the disease. They produce a robust immune response to the natural disease-causing virus. Generally, you only need a single shot of the live attenuated vaccine to establish protection. Inactivated vaccines are produced by inactivating the disease-causing natural virus either by chemicals or exposing to UV light. When given as a vaccine, inactivated viruses cannot replicate or cause the disease. These vaccines require booster shots or subsequent vaccinations as they are not as immunogenic as live attenuated vaccines. Viral vector vaccines utilize another virus that is harmless to humans to carry instruction from the disease-causing virus to produce individual parts or the disease-causing virus's proteins. For example, the S protein of SARS-CoV-2 virus. In this approach, the harmless viral vector converts our cells to manufacture SARS-CoV-2 proteins for our immune system to mount an immune reaction to protect us from the natural disease-causing virus. Some viral vectors can replicate within our cells, while others can't. RNA or DNA based vaccines are called nucleic acid based vaccines. They are also called genetic vaccines. These vaccines, either in the form of DNA or RNA, carry instructions to produce individual proteins of the disease causing, for example, SARS-CoV-2 virus. For example, instructions to make the S protein. These DNA or RNA molecules, when introduced as vaccines, convert our cells to manufacture the S protein of SARS-CoV-2 virus. So far, there are no vaccines produced by nucleic acid-based platforms for human use. However, things have already changed with the SARS-CoV-2 vaccine development, as the leading vaccine candidates are emerging from the mRNA platform. It would be difficult to predict the best platform to develop a vaccine against the SARS-CoV-2 virus. There are other critical factors in play as well. What age group benefits the most from the vaccine produced by which platform? Could the manufacturing process afford the global demand for the vaccine? What conditions are required to store the vaccine once manufactured till people get vaccinated? This has an impact on the worldwide distribution of COVID-19 vaccines. To control a pandemic, equitable distribution of a vaccine across all countries is a requirement. Then, the efficacy of the vaccine, the duration of the protection, and the cost for manufacturing will become critical components of the development process. We will not have information on how long the protection from the vaccine would last. The most important information is how safe the vaccine is going to be. So many questions may remain unanswered by the time a vaccine rolls out to people. All available vaccine development platforms are currently at work to produce vaccines for SARS-CoV-2 virus or COVID-19. This image shows some examples of organizations dedicated their efforts in developing a vaccine across different platforms. Traditionally, it takes a long time to bring a vaccine into the market for a new infection like SARS-CoV-2. It takes at least 5 to 10 years under a conventional approach with an investment of $1 to $2 billion to produce a vaccine. So far, the fastest vaccine ever produced took 4 years. That was against the Ebola infection. The high failure rate of vaccine candidates amounts to the high cost of development. The advancement of a vaccine into the clinic takes place through several different phases placed in a sequential manner as shown here. A candidate vaccine must fulfill specific criteria in the preceding phase before it is brought to the next phase. The vaccine development process initiates at the preclinical phase. Here, a development platform and the target of the SARS-CoV-2 virus that will be used to train our immune system are identified. Then the proof of concept studies are performed in cell culture followed by in animal models. Typically, non-human primates like monkeys are used to test the efficacy and safety of vaccine candidates. Once satisfactory results are obtained in the preclinical phase, the vaccine candidate will advance to the next phase, the clinical phase of development. This is initiated with the first-in-human studies in a phase 1 clinical trial in which the safety of the vaccine is evaluated in a small number between 50 to 100 healthy volunteers. Safety parameters include injection site reactions, headache, fever, body ache, nausea, vomiting, to mention a few. In addition to safety parameters, the immunogenicity of the candidate vaccine will also be evaluated. Immunogenicity means whether the vaccine candidate can activate the human immune system as measured by the generation of antibodies that recognize the viral target included in the vaccine. For example, the S protein of SARS-CoV-2 virus. Once the safety and immunogenicity profiles are acceptable, then the vaccine candidate will be brought to a phase 2 clinical trial in which several hundreds of human subjects of different age groups will be used. Again, evaluate safety and immunogenicity parameters. Phase 2 studies are also to identify the correct dose of the vaccine that will produce effective and acceptable immunogenicity with a reasonable and acceptable safety profile. Typically, the effective immunogenicity is considered the vaccine dose that produces blocking or neutralizing antibodies, in this case against the S protein in a natural infection. That is a neutralizing antibody level found in convalescent plasma of COVID-19 patients. In some cases, a booster or second shot of vaccine may require to achieve the effective immunogenicity level. Once the correct dose and dosing regimen with or without booster shots are identified, large phase 3 clinical trials with several thousands of human subjects will be initiated. The involvement of a large number of people provides an opportunity to observe any rare safety event that would have missed in phase 1 and 2 trials with a smaller number of people. Phase 3 trials are placebo-controlled and blinded clinical studies. Placebo-controlled means one group of people will receive a placebo or a dummy vaccine and the other group of people will receive the actual vaccine candidate. Blinded means the investigator does not know who received the placebo and who received the candidate vaccine. At the end of the study, an independent study group unblinds the trial results. Since phase 3 trials are long studies, sometimes an interim analysis of a phase 3 trial is conducted to analyze data in sample populations from both placebo and vaccine groups. This is to know quickly how well or how poorly the vaccine candidate is performing. It is also important to determine whether to continue the trial by enrolling new subjects or not. Having good outcomes of phase 3 clinical trial data with acceptable safety and efficacy, an application called BLA, Biologic License Application, will be filed with an appropriate regulatory body. For example, it is the FDA or the Federal Drug Agency in the United States and EMA, European Medical Agency in the European Union. A BLA is filed to request approval for the human use of the new vaccine. Typically, the review process of a BLA by these regulatory bodies take a long period of time, lasting around 1 year. Once the vaccine is approved, next comes another challenging phase: the distribution of the vaccine for vaccination. By now, you have a reasonable understanding of why it takes such a long time to develop a vaccine to get approval for clinical use using a traditional approach. For example, it took 28 years to develop a vaccine against chickenpox and 15 years for a vaccine to control the human papilloma viral infection. To control an ongoing viral pandemic like SARS-CoV-2, a vaccine must be developed quickly, in essence, to beat the rate of spreading of the virus. On March 11, 2020, WHO declared SARS-CoV-2 is a pandemic. As we stand now in November 2020, just after 9 months, we are about to have not one but several vaccines to combat the SARS-CoV-2 virus and the disease COVID-19. The immediate question that comes to our minds is how safe these vaccines are going to be. We must remember, a vaccine is given to healthy people and not to the sick. For this entire vaccination effort to reap its goal, a significant amount, at least 67% in each community, must be vaccinated in all age groups. The next question that lingers in our minds is that if it takes several years to develop a vaccine, why didn't the COVID-19 vaccine take that long and not even a year? By shortcutting the entire developmental process, are there risks associated with the final product, the vaccine? Several factors stack in favor of cutting down the timeline for developing a COVID-19 vaccine. First, several companies reaching the finish line of developing a COVID-19 vaccine had previous experience in the development process of vaccines against two other beta coronaviruses, SARS and MERS. Being a beta coronavirus, SARS-CoV-2 shares similar characteristics to SARS and MERS. The original SARS outbreak occurred in 2003. That infection was contained within 4 countries and finally extinguished the virus from human circulation before it became a pandemic. As such, the development of a vaccine against SARS virus was abandoned. MERS virus is still in circulation in Middle Eastern countries. Although it was initiated, the development of a vaccine against MERS was not completed. However, the good news was several companies already had experience in the initial phases of vaccine development against SARS and MERS, which gave them the privilege to hit the ground running with the development of a vaccine against the SARS-CoV-2 virus. It is unprecedented to test a vaccine candidate in humans just 65 days after publishing the RNA sequence of a virus. That is what the biotech company Moderna did with their mRNA platform to develop a COVID-19 vaccine. Second, regulatory agencies allowed the vaccine developers to stagger the phases in the overall vaccine developmental process. That is to run preclinical animal studies in parallel with first-in-human phase 1 clinical studies. Staggering phase 1 and phase 2 clinical studies is not that uncommon, and it happened here in the development of COVID-19 vaccine. Then phase 2 and phase 3 studies ran in parallel too. Having several staggered phases cut down the overall timeline of the vaccine development. A typical phase 3 trial in vaccine development is quite long, well over 1 year. In a typical phase 3 trial, after vaccinating with either the candidate vaccine or the placebo, then the human subjects are sent to a natural environment where an ongoing infection occurs. Data from interim analysis allowed filing applications to expedite the regulatory review to accelerate the approval process. Getting infected by SARS-CoV-2 in a natural community might not be fast enough for phase 3 trials to complete. There is still an option to consider a human challenge study within phase 3 to speed up the process. After vaccination with either the candidate vaccine or the placebo in a human challenge trial, human subjects are actively infected with the SARS-CoV-2 virus. Based on their altruistic gesture, close to 40,000 people from 166 countries are already registered for participating in human challenge studies. Then the regulatory review process is also taking place in an accelerated pace to complete within several weeks, certainly not taking a year. To achieve the hefty goal of developing a COVID-19 vaccine for the world in timely manner, several US government sectors joined together to initiate what is called Operation Warp Speed. It is a crucial contributor to this effort. To accelerate this unprecedented and ambitious goal, multiple candidate vaccines are being advanced through all available vaccine development platforms. Since 1 in 10 candidates has a chance to win the race, having numerous candidates on each platform will help. Once a vaccine is approved, it needs to be distributed globally without a delay. To achieve this goal, the vaccine manufacturing processes are not only already set up to scale productions, but also manufacturing of millions of doses is currently underway even before phase 3 clinical trial results become available. This is a substantial financial risk that companies are undertaking. If 2 shots per person are needed for a vaccine, its global demand will amount to 16 billion doses. Even 10 billion is an unimaginable scale. To meet such a demand, having more than 1 approved vaccines from different platforms would be helpful. The idea is to pick several top winners out of several vaccine candidates from different platforms. At the moment, there are at least 180 confirmed vaccine candidates, out of which 147 are actively tracking, 37 in phase 1, 17 in phase 2, and 13 in phase 3. To afford this enormous financial burden to develop a successful COVID-19 vaccine, several private and public partnerships had already been forged. The bulk of the burden is being shared by pharmaceutical companies. These efforts are multinational and spread across North America, China, Europe, Asia, Australia, and Russia. Approved vaccines must be manufactured quickly to a scale to meet the global demand. Furthermore, vaccines must be accessible to all countries. Most importantly, they have to be safe, efficacious, with a reasonable duration of protection. If the protection lasts only for a year, then the COVID-19 vaccine may be given concurrently with the flu vaccine on an annual basis until the SARS-CoV-2 virus is eliminated from human circulation. Currently, potential vaccine candidates are being manufactured to have a stockpile of inventory and are waiting to receive a green light from the FDA for their distribution. This highlights 5 leading COVID-19 vaccine candidates. The top 2 indicated in red are the mRNA-based vaccine candidates from Moderna and Pfizer-BioNTech. Upon injection, both produce the full-length SARS-CoV-2 protein in our body. Interim analysis of ongoing phase 3 trials revealed that they both have 95% efficacy, which is quite good. In fact, the FDA had set a low bar of 50% and above efficacy as one of the approval criteria for a COVID-19 vaccine. There are no 100% effective vaccine. However, the higher the efficacy of a vaccine, the smaller the number of doses needed to cover a population, and the shorter the time required to extinguish the virus from human circulation. These mRNA vaccines can be manufactured several millions to 1 billion doses per year. Novavax is a protein-based vaccine that contains already produced full-length S protein of the SARS-CoV-2 virus and formulated into an injection. Upon injection, our immune system immediately recognizes the viral protein to mount an immune reaction. So far, its efficacy data is not available. This platform can manufacture 2 billion doses per year. The AstraZeneca-Oxford University collaboration utilizes the viral vector platform used in a chimpanzee common cold adenoviral vector. This adenoviral vector contains the instructions in the form of DNA to produce a full-length S protein of SARS-CoV-2 virus when injected as a vaccine. AstraZeneca collaborates with the Serum Institute of India to manufacture the vaccine. There was a rush to report the efficacy from the interim phase 3 data of this vaccine that confused the public and scientists. The efficacy of this vaccine with an initial injection of the half dose followed by a month later with a full dose produced 90% efficacy. On the other hand, the efficacy was 62% when 2 full doses were given a month apart. Although several explanations have been offered, the exact reason for this observation awaits further research. CanSino Biologics is a Chinese company that collaborates with the Chinese military. In the recent past, this company introduced an Ebola vaccine. They use the same platform that they use for their Ebola vaccine, in which a human common cold adenoviral vector is stitched with DNA that instructs to produce the full-length S protein of SARS-CoV-2. Clinical data or the efficacy of this vaccine is not available. However, this vaccine is approved in China for limited use. In fact, CanSino Biologics was the first to test this COVID-19 vaccine candidate in humans on March 16, 2020, the same day that Moderna tested its mRNA vaccine. But CanSino Biologics was 8 hours ahead of Moderna. Several COVID-19 vaccines are already approved for limited use in 3 different countries: China, Russia, and UAE. In China, there are 2 vaccines, one from CanSino Biologics viral vector-based vaccine, and the second vaccine is from Sinovac, which is based on the inactivated SARS-CoV-2 virus. These 2 vaccines were approved in June and July in China for limited use. The next one was in Russia. A combination of 2 adenoviral vectors that were engineered to express the full-length S protein of SARS-CoV-2 were included in this vaccine, produced by Gamaleya Research Institute. This vaccine, called Sputnik V, was approved in Russia for limited use in August of 2020. The third vaccine was approved in UAE for limited use in September 2020. That vaccine was produced by Wuhan Institute of Biological Products, and it is based on inactivated SARS-CoV-2 virus. All these 4 vaccines were approved for limited use just based on their phase 2 clinical data. Based on the immunogenicity and tolerability of phase 2 trial data, several candidates' vaccines can be segregated. On the immunogenicity profile, CanSino Biologics' human adenoviral vector-based vaccine is at a lower end, while the Oxford AstraZeneca vaccine and the 2 mRNA-based vaccines are in the middle. The highest immunogenicity is associated with the protein-based Novavax candidate. On the tolerability profile, the 2 viral vector-based vaccines are the least tolerable, while the 2 mRNA-based vaccines take the middle. Novavax candidate shows the highest tolerability. Profiles of these leading vaccine candidates vary further when we look at their stabilities upon storage. This characteristic plays an important role when it comes to their distribution for vaccination. In that regard, mRNA-based Pfizer-BioNTech vaccine is the least favorite, as it requires super cool temperatures less than -70 degrees Celsius for storage. The next in line is the second mRNA-based vaccine from Moderna that requires -20 degrees Celsius for long-term storage. The Oxford AstraZeneca and Novavax candidates are stable between 6 to 8 degrees Celsius for long-term storage. To meet the global demand and to effectively distribute vaccines to all corners of the world, having several vaccines with different stability profiles play an important role. Even in developed countries, -70 degrees Celsius freezers are not that common. It is likely to have different immunogenicity and tolerability when these vaccines are tested across different age groups, and such data will emerge soon in the future. This is because individuals at different stages of life tend to have differences in mounting an immune reaction to a vaccine, as well as tolerating with different degrees of side effects. For example, children tend to have less tolerability towards the side effects of vaccines. Hence, lower doses of vaccines may be needed for children. Older adults tend to show less immunogenicity towards vaccine. For them, higher doses or different regimes of vaccination consisting of several boosters might be needed. It is also likely that the first set of vaccines in their initial formulations may be replaced later with different formulations. Or else, they might be replaced with entirely different vaccines that are currently behind the race but may have better safety, tolerability, and efficacy profiles. We have to stay tuned for all those developments. Thank you for listening, and I hope you got some clarity around the vaccines that will soon be coming to our neighborhood pharmacies and doctors' offices. Thank you.